this week. So we're going to start experiment 20. It's a 3-part experiment, 3-week-long experiment. So we're going to do the first part in lab this week. There is a report form for the first part that is due either this week for Tuesday labs or next week for Thursday labs. But we'll talk about it. It's not as bad as it sounds. Okay. So we'll go through what's involved with that. You'll have a quiz this week, and then next week your experiment 17 report is due. So I would highly recommend try processing your NMR data. As far as I know, all the lab sections ran. So process your NMR data for experiment 17 before lab this week. That way if you have any questions or problems or things don't look right, you can get that taken care of in lab this week instead of waiting until the 11th hour when things don't usually get fixed very well. So next week we'll do the second part of experiment 20B, and then, believe it or not, it's time to hand out the practice midterm exam next week. So I'll give that to you, and then on February 23rd will be our midterm exam. So with winter break all thrown in there, that throws everything off a little bit. So just don't forget, Monday, February 23rd, 4 to 5 in here is our midterm exam. Everyone will gather together, okay? I left the, I think Dr. Gilmore left this for me. Just a reminder, hopefully you saw this in whichever organic lecture you have, tomorrow at 7 o'clock in 1000, in the Science Center 1000, 3 students are going to be presenting information about research that they conducted last summer. And part of doing this is giving you more information about research groups and also giving you information about summer research as well. So on the bottom here is the SHARP deadline, which is the database that is used for submitting applications for research. So just keep that in mind, and if you're at all interested in summer research, this would be a very worthwhile experience for you to attend these lectures tomorrow night at 7. And the whole thing should take it like a normal seminar, hour-ish. Okay. All right. So we're gonna talk about our experiment that we're gonna look at for the next 3 weeks. So in this experiment, you guys are supposed to take on the role of technicians working for a pharmaceutical company. So we're working for Enzatech. It is housed in Zealand, so a nearby pharmaceutical company. And they have found that they've got some intermediates for a promising blockbuster drug that they can get from garlic, leeks, and onions. So, you know, they can get it, but it's not really the greatest thing to extract from those things. So what you have been given is the task to synthesize the intermediate that they desire and figure out what, under the conditions we're going to use, what would be the best way of synthesizing those intermediates, okay? So keep in mind that in the end, you're trying to fulfill the goal of what is needed by this pharmaceutical company, and you are in this process working for this pharmaceutical company. So what we are going to do is, in the first couple steps of the synthesis, let me get a little more clear here. First couple steps of the synthesis, and this scheme is in your lab manual. We're starting with 4-hydroxybenzoic acid, okay? And we want to make a diester using 4-hydroxybenzoic acid. So we'll do that over the course of 2 steps. Then we're going to go on and introduce your diester to an enzyme and see what happens in the presence of the enzyme, okay? And so in the end, what we want is selective hydrolysis from that enzyme, particularly this side, to be able to get towards that intermediate that Enzatech wants, okay? So what we're going to do is, in the end, there will be 18 possibilities of the 3 reactions that took place, okay? So there's 18 different sequences of reactions. And in the end, we'll pool data to look at what was the best sequence, and especially that best final step for doing what needed to happen, okay? So the first step that we're going to do this week is the step A, and we're going to take our 4-hydroxybenzoic acid, so everybody's starting with 4-hydroxybenzoic acid, and you are going to use either methanol, ethanol, or 1-propanol, and you're going to do a Fischer esterification, okay? And so then you'll get this ester, and then next week you'll go on and use an acid chloride. So we'll use our group here will either be methyl, ethyl, or propyl. With the acid chloride in the presence of pyridine, we'll make our diester, and then the week, not the week of winter break, but the week after winter break, then we'll do the last step with the enzyme, okay? So what I want you guys to take from, besides our quick synopsis of all of our steps, is here is kind of the big table that explains all of it, okay? So this table as well is in your lab manual. So you should have gotten a technician number from your lab prof last week, okay? Or hopefully, if you didn't get it last week, hopefully it's on the way, okay? Because you need that to know what you're going to use for your experiment as well as what you're going to use for the pre-lab for the experiment, okay? So you should have gotten a number between 1 and 18. And so then, depending on your number, you're just going to follow across the table to figure out what you're going to use each week, okay? So this first week, step A, if you have a technician number where there's a methyl group here, you're going to use methanol for your Fischer esterification. If you have a technician number where there's an ethyl group here, you're going to use ethanol. If you have a technician number where you have a propyl group here, you'll use 1-propanol. So those are the three possibilities for the alcohols, okay? And so you want to write your pre-lab specific to the reaction you're doing and specific to the alcohol you're using, okay? And I'll talk a little bit at the end about what needs to go into your notebook, but each week you're going to write a new pre-lab for each part, okay? So your pre-lab for part this week is just for part A, okay? You're not going to pre-lab all 3 parts. You're just going to pre-lab the first part, and then next week you'll pre-lab the second part, and when we get to the 3rd week, you'll pre-lab the 3rd part. So in case you want to work ahead between now and next week, I'll explain for step B here a little bit what each of these groups are pointing at, okay? So for step B, if your technician number has a methyl group, then you're going to use acetyl chloride. If your technician number has an ethyl group, you'll use propanyl chloride, and this is next week for part B. And then if your technician number has a propyl group, you'll use butyryl chloride. Okay, so if you want to work ahead on your pre-lab, now you know which acid chloride to use for next week, all right? So remember, we're doing step A this week. We're varying in alcohol. It won't be till next week we vary in acid chloride, okay? And then for the 3rd week, it tells you which enzyme you're going to use, all right? So we're going to look at the reaction that you're going to do this week, okay? And I'm going to use, so this mechanism is in your lab manual. You also are going to cover Fischer esterification when you cover carbonyl chemistry here in a couple weeks in lecture as well, okay? But if you want to look at it in your textbook, 7th edition is page 795, 796. 6th edition is 781 to 782, okay? So we're going to look at a couple things that are important about this reaction. First of all, it's an equilibrium reaction, so we need to keep that in mind when we're setting up the best conditions for it. So when you have an equilibrium reaction, what does that kind of tell you about as far as reactants and products? Anything? thoughts? No thoughts? Does it go like directly to product? You're going back and forth, right? And so how do you influence going towards products? How can you push it that direction? What do you want to do with your starting materials? Remember from Gen Chem? You want to kind of stockpile those, right? You want a lot of those around, so then it wants to go the other direction, okay? Versus if you don't have a lot of starting materials present, then it's going to want to hang out at starting materials as it makes product, okay? So, it's equilibrium reaction. We need to push it to the products. And we've got 4-hydroxybenzoic acid, we've got alcohol, and then we've got acid catalyst in the reaction, okay? So we can't really use an excess of the 4-hydroxybenzoic acid without running into like huge solubility problems and things like that, okay? It's a solid. So we're going to use an excess, a big excess, of the alcohol, okay? And so much of an excess that not only is this going to be a reagent, but this is going to be the solvent of the reaction. Okay, so we're going to use a lot of alcohol to push it to the direction of the products. Okay. Now, if you look down here, we're in the presence of an acid catalyst. Okay, so we end up basically hydronium ion as one of our byproducts. So basically protonated water, okay? We don't want to get a lot of water in our reaction because then we would be finding ourselves trying to go back the other direction, okay? So if we have too much of one of our products, it's going to want to go back the other direction. So what we're going to do is with our acid, it's hard to have acid that doesn't have a little bit of water in it, but we're going to use concentrated sulfuric acid so that the little bit of water that is in it, by using excess of alcohol, concentrated sulfuric acid, we're going to still have more success pushing it to the reactants, okay? So we're going to use concentrated sulfuric acid to minimize the water. So what does that tell you about how you want to set up this reaction and how you want to take care of things with your reaction setup and getting things going? You really want to make things as dry as possible. Really try not to add any water to the situation, okay? So use dry glassware. Don't try and add anything in there that's going to be a problem, okay? The other thing is be really careful with the acetone. We don't want other side reactions going on. So make sure you don't really want to rinse everything with acetone right before you start, because that could cause you some other problems with side products and so forth, okay, in the presence of the acid. So make things as dry as possible. We're going to use a lot of alcohol, and then we're going to use concentrated sulfuric acid. So with that, be careful with handling the sulfuric acid because it is concentrated, and make sure you wear gloves. Don't get it on yourself. If we drip a little on the hood, make sure we clean it up, okay? So what you guys are told in the initial instructions, you're given how much 4-hydroxybenzoic acid to use, but then you have to make calculations for how much alcohol you need to use based on that amount, okay? So make sure in your notebook, in your pre-lab, you show those calculations and make sure you've gone through those calculations correctly to make sure they're correct for your amount of alcohol, okay? And so what we've got here with this reaction is it is acid catalyzed, okay? So you could also potentially look at this in a base mechanism, but this works really well with this setup for acid catalyzed, okay? So we are first going to use our electrons from our oxygen on our carbonyl. That's going to help donate electrons to an acid proton. And so we form this charged intermediate that then, with the attack of the alcohol, we're going to have our electrons kick up as well. And so then we've got this tetrahedral intermediate that we're forming in the middle. And all the way through, we've got these equilibrium arrows, okay? So we got to do what we can to push it through all of these equilibria. So then we've got acid and we've got a little bit of water present, so we're going to deprotonate our OH group here that formed from our carbonyl, and then we're going to have a hydride shift too, because our water is a better leaving group than our alcohol, and it wouldn't work out so well if our alcohol left, because that would kind of undo what we just did. And so, or our OR group leaving. So then we reform our carbonyl, we've got water leaving, and so then we go on and form our ester, and then like I said, we've got hydronium ion left, okay? And so this is known as an acid catalyzed nucleophilic. So we're involving nucleophiles most of the time here, attacking our carbonyl. Acyl substitution, okay? So we're substituting for this OH group of our acid, the OR group of the newly formed, or OR prime group of the newly formed ester, all right? And this is from carboxylic acids are a very common starting material for esters, okay? So this is a pretty common reaction. Now, a couple things with this reaction is it does have some limitations, okay? So there is a reason that we're using the alcohols that you are going to use, okay? So primary alcohols are the best for this reaction. We go on and say that our secondary alcohols would be better than our tertiary alcohols, okay? But really, once you start getting into secondary, you'll get some reaction. When you get into tertiary, you're really not getting a lot of reaction with this, with what you've got going on here. You end up with other side reactions happening. So it's not the best method to making an ester if you're going to start with a tertiary alcohol, okay? And so we've set it up so that we're using all primary alcohols so we can get this reaction done in appropriate time. Yeah?
So is this SN2?
Pardon?
Is it like SN2 then?
Yeah. You could think of it as, and a lot of, you'll find when you cover carbonyl chemistry, a lot of it is kind of this similar setup, kind of. Yeah. So with the esters, with all of the types of carbonyl chemistry, they're going to go through this attack of the carbonyl, reformation of the carbonyl. It's all going to be similar type of chemistry. So anyway, we have prepared this reaction so that we can make things happen in a 5-hour lab for you, okay? But what we're going to need to do is a 2-hour reflux. So we've yet to do this yet, right? So we've gone 1 hour, but now we're going to push you to 2 hours. But what we're going to do is with that 2-hour reflux, where you're going to set up the reaction first. Okay, so when you come in to lab, you're going to set up your reaction. So make sure that you have your pre-lab all ready to go. You have your calculations already set up. You're all set to go for setting up your reaction. Okay? We're going to set up the reaction, get everybody refluxing, and then we'll take the quiz. Okay? So be ready this week that as soon as you come into lab, you're ready to go. All right? So especially 7:30 lab people will have to kind of get the cobwebs out a little bit and be ready to jump in and set up a reaction straight away, okay? Then, once everybody's refluxing nicely, we'll take the quiz, we'll go through other pre-lab instructions. And so, it is a 2-hour reflux, but we're gonna consume some of that time with stuff we would have done anyway. So, hopefully it doesn't feel quite like a 2-hour reflux, okay? And beyond what pre-lab instructions and other setup you guys need to do, don't you guys have an exam this week too? Yeah. So, be a good time to bring that stuff along too. You might have a few minutes for that as well in organic class. So then after the reflux, okay, and one key with this is we don't want to, another problem that you can have with this reaction is you really overcook things won't go so well, okay? We're heating everything with concentrated sulfuric acid. We don't want to quite overdo it, okay? So we are going to heat with a heating mantle, but get a gentle reflux going. We don't need a roaring boil reflux, okay? So nice reflux, not, halfway up the condenser reflux, okay? Then what we need to do is distill, after our 2 hours, we're going to distill the alcohol, okay? And the reason for that is we need to, alcohols, as you know, fairly soluble in water, and we're using small alcohols that are going to be pretty soluble in water, okay? You remember solubility tests from all the qual experiments we've done. So we need to distill off that alcohol so we get rid of the alcohol before we go on and start the rest of the workup, okay? So, methanol and ethanol, you're going to distill with a hot water bath. Okay, so kind of as best you can, convert from using the heating mantle or the reflux to then using a hot water bath to then set up a simple distillation, okay? But be careful, that heating mantle is going to be hot. We're heating it for 2 hours, refluxing it, okay? So handle it with a hot pad, kind of carefully lift everything up, get it transferred over to the back of your hood, and then get the water bath underneath things and get things set up for simple distillation, okay? So you want to not let things cool down too much, but be careful, don't burn yourself on the heating mantle as well, okay? The 1-propanol folks are going to use the rotovapes, okay? Which sounds, kind of unfair, but go and look up 1-propanol's boiling point, and in the end, it's going to be kind of like a tie here, okay? So 1-propanol has such a high boiling point, it's going to be hard for us to distill it off. It's better to use the rotovapes, but it's not going to be like your hexane ethyl acetate last week that it's just going to fly right off. It's going to take a little bit of doing to take the 1-propanol off. And again, we've got to be careful. We've got that acid in there. We don't want to overdo things, and we don't want to send things back the other direction and create impurities. So we've got to be careful with the heating here. We don't want, like 80 degrees on the water bath on the rotovac. Okay, so gentle heating here with the rotovac. Okay, so then we're going to go through a series of washes. Once we've got all of our alcohol off, or as much alcohol off as we can get off, then we're going to, and you're going to distill it as far as you can take it, and with the 1-propanol, rotovap off as much as you can take off, okay? So then with the rest of the workup, we're first going to dissolve the residue in ether. In ethyl ether, okay? So remember ethyl ether, low boiling point, we've got things pretty hot, let things cool to room temperature, then we're going to add the ethyl ether, get everything dissolved, okay? Then what we need to do are 2 washes with water, okay? And remember, when we're talking washing, remember this involves separatory funnel. So we're going to dissolve in ether, put our dissolved ether mixture in the separatory funnel. Make sure you don't end up draining out the bottom because the stopcock's open. Then you're going to add 25 mils of water, put the stopper on it, shake and vent, shake and vent, shake and vent, drain off the water. That whole process was one wash, okay? So you're going to do that twice, all right? And the whole reason for this is to help remove the bulk of the sodium, or of the sulfuric acid. Okay. The next thing we're going to do is neutralize the rest of it with base, but before we do that, we want to get as much of it out as possible. So with these washes, you're going to have acidic washes, so be careful handling them, okay? Don't get them on you. Wear gloves while you're doing the washes, okay? So then we're going to do a 25 milliliter wash with sodium bicarbonate. That's going to remove what little residual sulfuric acid is left. And what else do you think it could remove from our process here? And think about not so much the generic reaction, but think about what our starting material is. So this is where we're going. This is our starting material. So it's going to deprotonate which end of it? The acid, right? Because we don't really want to deprotonate the phenol, then we're in all sorts of trouble because then we're going to get rid of our ester and our carboxylic acid. But it will, sodium bicarbonate is basic enough to deprotonate the acid, and so it's going to help remove the residual starting material. And so the other thing with this reaction is the methyl group. Methyl groups are easy to put on. It gets a little harder to put on. Ethyl groups gets a little harder to put on one propanol group. So as long as the chain gets bigger, it's also harder to add those on. So one propanol people are probably going to have more residual starting material than the methanol people are going to. Okay, but we need to get it out of everybody's before we go on to the next step. Okay, so with experiment 20, we're not going to do a column along the way or anything like that to clean things up. We've got to clean things up in our workup. Okay, so that's why we're going to do the bicarbonate wash. Then we're going to do a wash with saturated sodium chloride or brine. Okay. And so that's going to help remove residual salts. And it also helps with, we've got solvent is ether. We've got salty water that's not going to be soluble with the ether. It's also going to help draw out a little bit, helps dry the ether a little bit. Okay. Now it's not going to take all the water out, but it does help take a little bit out, okay? Then we're going to dry with magnesium sulfate. Okay, so if you remember that from first semester, I'm going to add that to our ether solution. Remember, you're looking for the snow globe where you've got a little bit of clumps and a little bit free floating. Swirl it around, make sure you've got enough mag sulfate in it, but not too much, and then let it sit 5-10 minutes to dry. And then how do you usually filter away magnesium sulfate? gravity filtration, right? So when you want the filtrate, you usually use gravity. When you want the solid, you use vacuum. So we want the filtrate. One thing to be careful with, though, is follow the instructions for rinsing that mag sulfate with ether. Your product, and it doesn't matter which alcohol you use, your product and the mag sulfate are going to look really similar to each other, okay? So it's going to be hard to tell, is that mag sulfate just mag sulfate, or did a little bit of the solid crash out because some of the ether evaporated, okay? And especially people putting on the methyl group, that crashes out of ether really easily, okay? So you're going to need to rinse that mag sulfate with ether to make sure you get all the solid off the magnesium sulfate, and you don't by accident throw away all your product when you throw away the magnesium sulfate, okay? So be careful to follow the instructions for rinsing, and make sure that you aren't leaving any solid behind that you want, okay? And then that mag sulfate can go in the bucket in the, once it's dried out a little bit, it can go in the bucket in the reagent hood with the filter paper, okay? So then, now everybody gets to rotovap, okay, to remove the ether. Now, something really helpful anytime you're rotovapping to get to your final product, always know the tare weight. So we talked about this last week. Know the tare weight of the round bottom flask that you're using for rotovapping. Okay? And how full can you fill a round bottom flask when you're rotovapping again? Half full, so not more than half full. So approximate how much ether you've got left to figure out which round bottom to use. So then, after you rotovap, and it's ether, it's going to fly off of there really fast. Do be careful. We don't want. So once everybody's taken off their propanol, we need to cool those baths down again, the water baths on the rotovaps, because the ether is going to zoom right off of there. Okay, so you don't need a lot of heat with ether. Also, control the vacuum. You don't need a ton of vacuum either to pull off the ether. Okay, so kind of pay attention to what you're doing with the rotovap. We'll put it on the high vacuum pump. Okay, and then you first of all want to figure out yield. Okay, so if you know the tare weight of your round bottom after the high vacuum pump, you'll know what your yield is. Make sure you record this. This is really important. Then you're going to set aside about 0.1 grams of it for TLC for parts A and B, okay? Or sorry, parts B and C, all right? So I put this in a vial because you're going to need it multiple times, okay? Now hopefully with your yield, you don't only have 0.1 grams because you don't want to set aside all of your material if that's all you've got, all right? Because we need 1.5 grams or so for next week, okay? So once you get your yield, if you're really running low on it, talk to your lab prof and see how much of this you should pull aside, okay? But if you're really careful following through, especially with the workup and rinsing your mag sulfate, hopefully your yield is good enough that setting aside 0.1 grams is okay, all right? Then you're going to need prepare an NMR sample, and remember you need 5 to 10 mgs, so that's not very much. And most of you at this step will probably have solids, so if you need help preparing those, we can help you prepare it with a solid. So 5 to 10 milligrams, which is 0.005 to 0.010 grams. And then you need to collect an IR spectrum. So So at the end of all this, you want at least about 1.5 grams left. Okay. And we're going to run your NMR samples right away as soon as we can, because then we need to get ready for the report form that you need to hand in. Okay. So any questions with this before I go through things that need to be in your notebook, and then we'll look at the report form. Good with the procedure? Good with what alcohol you're using? Okay. So for your notebook, for all of these steps, we've already kind of talked about pre-lab. So you're going to have a pre-lab for each step. Okay, then you're going to have experimental for each step. And then you're going to have something we haven't really written before, but maybe you kind of have for your fall experiments. You want a mini conclusion for each step. Okay, so this is not the full-blown kit and caboodle. And with that, you're going to collect IR data and NMR data along the way, so you make sure that you have IR and NMR tables in your notebook. Okay? And for a little bit here, your spectra is not going to be attached because it's going to be attached to that report form, which I'll talk about here in a minute. Okay? So with this mini conclusion, like I said, it's not the full-blown conclusion for that step. You just want to, in a couple paragraphs, summarize the step and summarize the data that was collected. And in that summary, you want to be discussing, can you move on? Keep in mind, the product that you make this week, you need to be able to move on to Part B with next week, okay? So in that summary, you also want to make sure you address, is the product that you made this week sufficient for moving on to Part B, okay? Or are there like huge impurities, or it didn't work, or things like that that need to be considered before you take that on to next week's experiment, okay? So with this mini conclusion, you should be writing this, so don't wait until experiment 20 is due. You should be writing this before you start the next step, okay? So all of these guys should be completed before you, for Part A, before you start Part B, all right? So make sure that mini conclusion is in there, even though we're not gonna necessarily check your notebook for it next week. Make sure it's in there before you go on to the next step, because it's really easy to actually figure out if someone has put it in there or not before you go on to the next step. And this can be, like, this doesn't have to be a word process, this can just be handwritten in your notebook, okay? Couple paragraphs, not anything lengthy, all right? So then, with that, we've got our report form here that sounds worse than it really is. So that is on page 2013, I believe, of your lab manual. Okay, so at the end of experiment 20, there's a report form for 20A and a report form for 20B. There won't be one for 20C. We'll talk about what we're going to do for that step when we get there. Okay, so this is worth 25 points. All right. There's not a whole lot to fill out. We'll go through the parts here in a minute. But big part of your grade for this is your analysis of your NMR and your IR. Okay? So do a really good job. You want to analyze those well. Make sure that you are making a table for both NMR and IR and attach that to this form. Make sure you attach your NMR spectra and your IR spectra to this form, okay? But keep in mind, your analysis of IR and NMR is a big part of your grade here. So there's not a ton to analyze with it, so if things, you know, don't get analyzed well, a bigger part of your grade will be marked off for that. So do a good job with your analysis, all right? So the first part with the IR spectrum, you're going to write down what peaks of the IR spectrum of your 28 product have changed from that of the starting material, okay? And so at some point tomorrow on Moodle, I am going to have an IR spectrum of 4-hydroxybenzoic acid taken with our IRs put up on Moodle. That is the spectrum that you're comparing it to for the starting material, okay? So I'll have that up on Moodle. Then you need to assign the peaks and make sure that you have a table that includes your experimental peaks from your IR spectrum and the peaks from the standard or the starting material for the 4-hydroxybenzoic acid, okay? And if you want to go on and find literature IR spectra, you could look for those as well. But you, at the least, compare from your product to the starting material, and hopefully you see a difference in there and differences that make sense, okay? And so on your IR spectrum itself, besides the table, make sure you label the peaks on that spectrum, all right? And kind of forgot here, fill in this information, draw the structure of what you made too, okay? And make sure your yield is really, really clear, because we're going to be looking at that as well. NMR, you're going to draw the structure of your compound on your NMR spectrum, label the unique protons with letters, label the peaks on the spectrum that correspond to those letters, okay? So make sure it's really clear what assignments you're making with your NMR spectrum. And if you're not sure what peaks you're going to assign or how they should be assigned, make sure that you use calculations and things like that to help you out, okay? So there aren't a ton of peaks in the product, so it's not, you know, if you miss one or two, it's not going to help your grade out a whole lot, so be really careful with that. Then you're going to also, with that NMR spectrum, make sure you label any impurities in there, okay? So what things do you think could be in there?
Water.
Water, potentially. Hopefully you get most of it out, but it could be in there. Acetone, if you're not careful with your NMR sample prep. Starting material, yep. If we don't get rid of all the 4-hydroxybenzoic acid, that's definitely a possibility. What was that?
Sulfuric acid.
Most likely you should be able to take care of that, but yeah, sulfuric acid is something that's really hard to see in the NMR, so probably won't see a whole lot of that. Ether, definitely, big possibility. And what's another one that you start with? There's a, yeah, if you don't do a good job distilling that alcohol, it still could be present, especially with the 1-propanol. So make sure to do a really good job rotovapping. Methanol and ethanol do a good job distilling, because it could still be there in the end, okay? So make sure you identify the impurities. As far as the ether, the water, the methanol, the ethanol, you can use that impurity table that I put up on Moodle. If you have 1-propanol, you'll probably want to look up that spectrum so you can match up those peaks. And then the 4-hydroxybenzoic acid, you'd have to figure out where those peaks are, okay? But hopefully it'll be easier to figure out, because the peaks to your product would be large and the peaks to impurities would be small, okay? With that, you probably want to also have a gauge for about how much of the impurity you have, especially if you have a large amount. And so remember, with NMR spectra, when you're looking at ratios of protons from two different compounds, you're not going to be comparing grams to grams. Remember, you're looking at mole ratios, okay? So you'd have to keep in account the mole ratio and then go on and convert those to grams to figure out how much of your weight is a certain impurity, if it's a significant impurity, okay? And then what evidence shows that you have the right product? And then you'd answer here as far as the impurities, are there any peaks? If so, then you want to list the chemical shift and what those peaks are. And in here also address, if you could go on to the next step, or if there's enough things going on here that maybe something needs to happen before you keep going, okay? So the reason for the tight deadlines for these guys, and you're going to have the report form, your IR spectrum, your NMR spectrum, your IR table, NMR table. That's what you're turning in with us. So it's not a ton of stuff that you're handing in. The reason for the tight deadline is because we need to look at this in time for you to do the experiment next week. So we need to look at it, and if there's something that needs to happen, we need that to be able to happen in Open Lab before you go on and take on Part B. Okay, so we need your Part A product to be good and clean and ready to go for Part B. So that's why we need to get that information so we can look at it and give you feedback. Yeah, you're good to go. No, we need to quick take care of this before you go on to the next step. Okay? So for as far as due dates, Tuesday lab, they're due on Friday for report forms. And the Thursday labs are due on Tuesday, so next week Tuesday. Okay, so this is 1:30. This is 2-3, okay? And as soon as we get them, we're going to immediately look at them and give you guys feedback. Most likely probably email you. If you don't hear from us, that usually means you're good. If you do hear from us, then that means there's something we need to take care of before you go on to the next step. Okay, questions at all with any of that? All right, and as soon as we get your NMR samples this week, we'll run them for you so that they're all set for you to go ahead and process and get that data. Okay?